So, good afternoon or evening, everyone. So my name is Dr. Harley Robertson-Cocks. I'm a lecturer in biomedical sciences at Newcastle University. So I'm in the non-clinical side of the medical school. But I have a bit of experience in the medical side, which I'll come on to. But I'm going to talk to you today about computational modeling of the heart. So my background is a bit of a weird mix in that I come from the kind of sciencey side, but have transitioned into the computer sciencey side as well. So basically what I do from a research side is develop computer models that simulate electrical activity of the heart. So you can see this diagram on the right-hand side over here um, is an ECG. Some of you are probably familiar with ECG, so the electrical activity of the heart. So I just want to start off first of all by saying if your ECG actually looks like this, I highly recommend that you go and see your GP because that is not normal. It's just a fancy little graphic for me to use for the slides. So a little bit about me first of all, because I just thought it'd be maybe a bit relatable to some of you that are applying to medicine to hear a bit of my background. So I'm from a small town in Lincolnshire, not very interesting really. Small, uh, well, a village really that I'm from, near a small town in a county that's otherwise farmland. So there's not a lot going on in Lincolnshire. And like a lot of students at school, I didn't really get a lot of good careers guidance. So there wasn't really a lot of support in terms of, when you're 15 and you start to think about what you want to do when you leave school. The best we got was an online quiz that we were allowed to do once that apparently tells you your ideal job. Mine came out as an epidemiologist. I'm not an epidemiologist, but funnily enough, nowadays that's actually quite a good match for the kind of skills and things that I'm interested in. But the actual guidance in place wasn't much good, in my opinion. I was relatively clever, I suppose, and I did like the human side of biology, so very much the human biology side. And that obviously meant that the school jumped on me and said, "Oh, we've got a clever student here who likes biology. He's got to go to medicine because that will look good for our stats." So I was kind of moved towards the medical area. And to some extent, I felt a bit of pressure to do a degree that kind of obviously led to a job. So in my mind, oh yeah, do medicine, become a doctor. That's straightforward. Whereas some degrees don't necessarily have that clear link. So even though I didn't necessarily have, a long time passion in medicine, I did think it made sense for my field at least. So it was a bit of a late decision, really. Unlike a lot of you who are probably going through and getting as much work experience as you can and things like that to support your applications for medicine, I didn't have any, which already put me on the back foot to some extent. Funnily enough, the area I'm from is a bit of a military area. So the Royal Air Force in particular, my granddad was in the Navy. So there was a part of me with a bit of pressure from my dad who said, well, maybe actually you should do medicine and then go and enter and join the RAF as a medical officer. So I had these grand aspirations, basically, at that stage. So a little bit more about my kind of background is I did biology, chemistry, and geography at A level. I liked geography, so I kept that in there. Um, and in terms of medical schools at the time, I applied to Leeds, Sheffield, Durham, who don't have a medical school anymore, I don't think, and Imperial College in London. So applying to Leeds, I got rejected within a couple of weeks of applying, without even getting invited to interview. And a few weeks after that, I got rejected from Sheffield as well, again, without even getting an interview. So it wasn't really the best of starts for my medical career. Really late in the application cycle, I actually got invited for an interview at Durham. So we're talking February, which is really quite late on for the normal admission cycle, or at least it was back in the day. Um, and then, funnily enough, they offered me a place, which I thought, oh, great, at least I've got one offer. And then, strangely, I got an interview invite at Imperial even later on in the cycle in sort of March, April time. And they offered me a place as well. So, weirdly, I had very different interview experiences at both of those. At Durham, I had a full 90-minute grilling with two interviewers who really took me to task on why I'd applied to interview, lots and lots of scenarios, what I would do if this circumstance happened, and things like that. So it was really quite intense. My interview at Imperial, which, considering it's quite a well-known and reputable university, was completely different. I basically spent 15 minutes talking about rugby with a panel of four interviewers. And then at the very end, they were like, oh, we should probably ask you a medical related question. What would you do about obesity if you were in charge of the government? And I gave some kind of brief answer about that. So the interview experiences were completely different, and yet somehow both led me to get offers. Now, like I said, I'm from a rural part of town, from Lincolnshire, so I was kind of dazzled by the bright lights of London. So I decided that I would accept the offer at Imperial. Um, which maybe wasn't the best of ideas, basically, because I didn't enjoy my time at London at all. I didn't enjoy the lifestyle of being in London. Personally, I didn't really enjoy the lifestyle of being a medical student, and that combination just didn't suit me at all. So made the time that I spent in London, which was just a year, not the best. I had a good time to some extent. So a couple of pictures of me on screen. First of all, in some scrubs at some Freshers Week social, probably pre-beard, and also when I had a good head of hair. And a picture here in the middle of actually me playing in what is actually the oldest rugby cup competition in the world, apparently. So the United Hospitals Cup is an ancient medical school rugby cup, and I got the chance to play in that. Basically, if it weren't for that part of the social life side of my time in university, I think I probably would have dropped out a bit earlier. But as it happened, I ended up seeing the whole year through. I didn't pass all my exams, which I'm not afraid to admit. So I ended up at the end of the year with the university saying, sorry, but you're going to have to leave. So again, plans got kind of scuppered to some extent. Now, at the time, no other universities had space for me to go and get in through clearing or something like that, because this was actually the year that the tuition fees went up. So there was an already high volume of applications to all the different universities, which meant I had to take a year out, which wasn't in my plans at all. Uh, for whatever reason, there's a company called Interflora who sell flowers, and their headquarters was in that small town near where I grew up. So I was lucky enough to get a job that kept me afloat for that year, basically selling flowers over the phone, or more, what was most likely the case, getting shouted at when people's flowers didn't turn up that they'd ordered. So I worked in the kind of customer service side, which, yeah, was a bag of laughs, shall we say. That allowed me to kind of reassess anyway. So I realized that I still had that interest in human biology from a basic science point of view. Um, so I decided to reapply to university, but not for medicine, and for kind of human biology related degrees. So I had offers for a range of sort of physiology, biomedical science related courses. And it was the human physiology program at Leeds that I was drawn to, and is what I ended up doing in the end. I really loved my time at Leeds. I spent a long, good 8 years in Leeds. Really great city. And here's a picture of me being the subject of one of many exercise physiology experiments, one of which was cycling for a couple of hours, I think, in 40-degree heat, which wasn't very pleasant. There was an open day that day as well. I didn't realize this at the time, but it meant that there were prospective parents and students walking around, looking at, getting a tour of the lab and things like that. Now, I didn't know that was happening, and I stepped out partway through the heat chamber experiment, absolutely dripping with sweat. And I kind of stepped out to catch my breath a little bit, during which time I was also swearing because of how hot it was. And I basically ended up flicking sweat off myself onto a semicircle of parents and students, which probably wasn't a good impression for them of the university experience. But yeah, it sums up how kind of physiology was really interesting to me, something that I really enjoyed. And then it was in my last year of uni that I started to think quite like to keep learning. Maybe a controversial opinion, but I quite like learning. So I applied to do some PhDs rather than go into masters, because I quite like the idea of kind of doing your own research and being able to forge your own path. And I was lucky enough to stay in Leeds, where I already was, and study for a PhD, for which I basically developed these computational models that I'm going to come on to talk about in a little bit. Now, while I was doing my PhD, I also got quite a lot of opportunity to teach. So to teach undergraduate students. And it was then that I kind of realized that actually I preferred the teaching side of things to the research, funnily enough. Um, so that was one thing. After my PhD, I worked for a brief time as a postdoctoral researcher. Still in the cardiovascular science field, and basically continuing a little bit of the work that I'd done during my PhD. But that was only ever going to be a short-term job. And that job ended in April 2020, which those of you with a good memory will realize that's probably not a very good time to start looking for a new job because that was just before COVID came along. Um, so loads of the jobs that I'd applied to after my postdoc basically got pulled because there was no point anyone hiring anyone while COVID had put a stop to everything. So the pandemic had really scuppered my plans a little bit more. Luckily, one job that I had applied to at Newcastle University was still hiring, and that's because it was just a short-term maternity cover role. And obviously, the member of staff that was going on maternity leave was going to be on maternity leave anyway, and they obviously needed somebody to come in and basically cover their workload. So I was fortunate enough to be offered that role, and that's where I've been since. So it went from a temporary contract to thankfully a full-time one because I managed to convince the right people, I suppose, that I was kind of enjoying my job and did a reasonable job that they wanted to keep me on. Starting a new job right at the beginning of pandemic in a different city to where I'm actually living was challenging. My first job was to try and figure out how we're going to try and teach the cohort of students that were due to start in a few months' time, considering everything that was going on. Um, but I'm still here, so it must have worked out all right. So a bit about my job. I'm teaching focused nowadays. I don't tend to do much research, and I'm mainly interested in teaching physiology. So my job title says biomedical sciences, but I identify as a physiologist, and mainly in the teaching side of things. So I've got a bit of physiology that I teach, a lot of cardiovascular science, but I also do a lot of stuff for our students in terms of wider academic skills like handling data, scientific writing, and things like that. Quite a lot of my time is spent in the lab, so doing practical classes. So I've got a couple of videos here which I hope will play okay on over Zoom. So this video I'm going to show you is basically one of the practicals we do in first year where we get students to electrocute each other. So it's quite fun. But by electrocuting different regions on the arm, they stimulate the ulnar nerve, which is a nerve that innervates one of the muscles in the palm of your hand. And in doing so, you will hopefully see in this video that it elicits that nice twitch of the hand, as you can see, hopefully. So what we get the students to do is electrocute themselves on the wrist and then do the same thing but on the elbow. And basically by recording the difference in time it takes for a response to be measured at the wrist and the elbow allows us to basically calculate the speed at which nerve impulses are traveling down the arm. So around about 70 meters per second is on a good nerve conduction velocity, we would call it. So it's quite an interesting lab, quite enjoyable one to get involved in. Few other roles at Newcastle as well. I'm interested in student employability, so students making the most out of their degree, going on to get the career that they want. So I help our students with stuff like that. And I also run our school Instagram account, so School of Biomedical, Nutritional and Sports Sciences, or NCL_BNS, if you want to give us a follow. We occasionally post serious stuff, but every now and then, basically, I like to just share some daft memes about science, like you can see on screen here. Um, just keep the students kind of engaged, I suppose. I'd imagine most of them are scrolling through Instagram while I'm lecturing them these days, but at least it keeps them on the science mind ticking over. So, on to my research. This is my thesis, a PhD thesis. It's about 250 pages long. In a PhD, you basically spend an awful lot of time writing a book, essentially, about your research. So my research, my thesis was called Biophysical Modeling of Wrapped Cardiac Electrophysiology and Calcium Handling. Now, that's quite a mouthful. Many of you are probably sat there thinking, I don't really know what that means. So I'm going to break it down for you. So, biophysical. Basically just means a mix of biology and physics. So that's all that really relates to. When I say modeling, I mean using computers, so computational. A rat is a long-tailed rodent, which I'm sure you all know. Cardiac means relating to the heart. Electrophysiology means the electrical properties of cells and tissues in terms of biology. And calcium is an important ion in the body. It's useful for bones and stuff like that, but it's also got a really important role in cells that contract like the heart does. So the long and short is basically that means writing computer programs that simulate electrical activity of the heart, basically. So the reason that's important, cardiovascular disease is one of the leading causes of mortality across the world. About 25% of deaths in the UK are believed to be thought to be due to cardiovascular disease. And the incidence of cardiovascular disease is only going up as we get a generally older population. It's estimated that there's twice as many people living with cardiovascular disease as Alzheimer's and cancer combined, but you hear quite a lot about neurodegenerative diseases like Alzheimer's and also cancer, but maybe less so about cardiovascular disease, despite the fact, how prevalent it is. Um, Here's some stats from the British Heart Foundation to give you an idea. So these are the most recent statistics they provide. So apparently, on average, today in the UK, 480 people will die from cardiovascular disease, as an example. So it really is um, a big, a big problem, if you like. Cardiovascular disease itself is a broad term, so it's an umbrella term for a range of different conditions that affect the heart and the blood vessels. So some examples of those are things like coronary heart disease or heart attacks, stroke, etc. The two that I'm most interested in are here in bold on screen, so that's arrhythmias or abnormal heart rhythms and heart failure, which I'll go on to explain in a little bit. So, heart failure itself is the inability of the heart to pump enough blood to meet the body's demands. Arrhythmias are just abnormal heart rhythms. So if you think about the fact that your heart beats roughly once per second, an abnormal heart rhythm would be that it's not beating once per second, it's beating irregularly. An example of that might be something like atrial fibrillation, which you may have heard of before. Um, Patients with heart failure can vary. So early stage heart failure might just be that when they're exercising quite vigorously, they start to struggle quicker than they should do for their levels of fitness, for example. But those with severe heart failure might struggle to do things like get off a chair, tie their shoelaces, go to the fridge, pick up a drink, stuff like that. So we're talking really severely impacted in terms of their cardiovascular system. I'm not going to talk through this diagram too much, but it just shows and conveys that heart failure and arrhythmias like atrial fibrillation are quite closely linked together. So one can cause the other and the other can cause the other, etc. So they're quite tightly interlinked, and the cardiovascular system is quite complex. Cardiovascular disease doesn't necessarily just have cardiovascular origins. These issues could be because of neural conditions or things like that. So it does get quite complicated, but I'm not going to go into that level of detail for now. So some of you might be thinking, right, but why do we do computer modeling then? What's the point? Why don't we just test on humans or do our normal experiments that we've done over, decades? Well, the problem is physiological systems and functions act across a massive range of time and length scales. So you might have molecular structures like genes, which could be nanometers in size. Those genes influence the activity of cells, which could be microns in size. Those cells determine the function of an organ, which might be sort of millimeters or centimeters in size. And then the whole body is, 1.8 meters tall or thereabouts. And on top of that, you've got all of the different time scales involved as well. So, you know, cells operate on like a second by second basis, or an example could be the heart beats every second. But as a human, we live for 70 plus years. So there's that vastly different time and length scale that influences how the body actually works. Now, while it's straightforward for us to look at any of these components individually, it's not so easy to see how those differences change from the different scales. So it's very different, very difficult, sorry, very costly probably to even try to study each of these um, collectively from an experimental point of view. But computer models nowadays allow us to do that. So I keep throwing this term around modeling. First thing that you're probably going to think of when I say that is some good-looking fella who's a model, and I'm not talking about that, obviously. You might have come across weather forecasts and things like that. That's an example of scientific modeling, but again, that's not what I'm after. Um, Architects might develop these kind of scientific models where they're measuring whether a structure is viable and stuff like that. Again, scientific form of modeling, but not what I'm on about. Formula One engineering teams will do all sorts of fancy science, modeling the flow of air and things like that across the vehicles to measure the aerodynamics. Again, also not what I'm talking about. So I'm talking about the kind of modeling where we reproduce things like the electrical activity of the heart to see how these arrhythmias might develop or how heart failure might impact them, things like that. So this video that you can see on screen is an example of how a computer model has been used to simulate the spread of electrical activity across the ventricles or the lower chambers of the heart. So that's what you can see there. Now, a quick history lesson. Um, These two gentlemen on screen, Alan Hodgkin and Andrew Huxley, were awarded the 1963 Nobel Prize in Physiology or Medicine, basically in recognition for the kind of fundamental work they did in early computational modeling, shall we say. So, hopefully, most of you are aware that cells have membranes, and embedded within those membranes of a cell are special proteins, which can act as transporters of molecules or ions. And in cells like skeletal muscle or nerves or cardiac muscle, in my case, either side of that membrane, there's usually an unequal distribution of ions, so things that are positive or negatively charged. So there might be more positive ions on one side of the membrane than the other, which means there's a difference in charge across that membrane. And these proteins might allow for these ions to flow into the cell, to flow out of the cell, or maybe a bit of both, as that animation shows. So what Hodgkin and Huxley proposed was that because the membrane of a cell is separating charge on either side, what it's basically doing is the same thing that an electrical circuit does. So an electrical circuit fundamentally is separating charge. So this thing on screen here just shows you an example of a simple electrical circuit. So we can say that a cell could be represented as an electrical circuit in which you've got charge being separated by a capacitor, as you can see over here, and you've got the flow of current across that membrane, i.e. the movement of ions, controlled by these variable resistors that you can see in the middle, which basically represent these special proteins. So because you can basically say, well, the cell is acting like an electrical circuit, that means that you can apply some maths to that physical principle. So you can basically derive some equations that calculate the flow of electrical current across that circuit, therefore the flow of electrical current across a cell membrane. And thankfully nowadays we don't have to do those calculations by hand. We've got computers that can do it all for us if we write a bit of code. So this is just a simple screenshot of some code that I might have written at some point. Now, back in the day when Hodgkin and Huxley were doing their work, they will have used a computer like you can see on the screen here, which doesn't have a screen. So they will have basically had to operate this as a glorified calculator where they will have been inputting bits of card with holes punched in it, and the position of the hole punches determine the calculations that they're performing. So very, very complicated. Thankfully, nowadays we have evolved technologically speaking. We've got things like the Newcastle University has a high performance computer or a supercomputer, which can do these calculations much, much faster and can do thousands, millions of them all at once as well. So we've got a much more improved technology. As an example, It might have taken Hodgkin and Huxley 2 hours manually putting stuff into their computer to calculate one heartbeat's worth of electrical impulses or electrical activity. But on a supercomputer, that would basically be instantaneous. In fact, if you could do it on your phone, you'd be able to have answers immediately. Even a simple desktop computer has the capacity to do these calculations much, much quicker nowadays. Now, don't get bogged down in the detail here, but this slide basically just shows you that what they developed 60, 70, 80 years ago was quite good for its time, basically. So it's the first major attempt at using maths to explain physiology, and it did a good job of reproducing a complex phenomenon, which is the action potential in the nerve. So that's this thing that you can see down here. So the change in electrical activity with an impulse in nerve cells, fundamentally. So if you look closely at the graphs that you can see on screen, basically you can see these dots and these solid lines. Now the dots represent the data they collected from experiments, and the lines represent what their computer model or their mathematical model predicted. And you can see fits it pretty much bang on. So that shows you that even though we're talking 70, 80 years ago, these were really high fidelity experiments and a model that does a good job of predicting the nerve action potential. Now, I'm a cardiovascular scientist, as I said. So there is this gentleman called Dennis Noble who basically 10 years later took what Hodgkin and Huxley had developed and adapted it to go from nerve to the heart. So this is his attempt at reproducing the cardiac action potential or the change in electrical activity in the heart, which looks slightly different, basically, is all you need to know for now. But again, based on that fundamental work. Now, you might be wondering if it's still useful because I'm talking, a long time in the past now. The answer is yes, it is. We wouldn't be where we are today without that fundamental work back in the 50s or the 40s and 50s by Hodgkin and Huxley. So we have much more detailed models nowadays, but in many ways, they're still reliant on the same principles that Hodgkin and Huxley developed back in the day. We know more about physiology as well, so we've discovered extra things like the calcium current in cardiac muscle, so the movement of calcium across the membrane. And nowadays, computer models are just really powerful tools. They can do stuff that an experiment can't, but ultimately, we work hand in hand between computer models and experiments to advance the kind of scientific field. So there's something what we call the virtuous circle, which is what you can see on here, where we develop these hypotheses, we do some experiments, that gives us some data from which we can build a model. That model gives us some data, we check that it matches the experiments, and this circle keeps going, and it helps us to advance the scientific field. So now I'm going to start getting into the substance of my research background. To do that, I need to give you a bit of a crash course in cardiac physiology. So it's a slightly complicated diagram that you can see on screen, but I'll talk you through it. What you're seeing here is a schematic of a heart muscle cell, basically. So the outer blue lines that you can see here represent the cell membrane. These coloured sort of shapes that you can see are different proteins, essentially. And then inside the cell, you can see these structures here as well. So this one here is called the sarcoplasmic reticulum. That is an internal store of calcium. These structures here are the myofilaments. They're the things that do the contraction. So when the muscles contract, those myofilaments move, essentially. This is your mitochondria, well known as the powerhouse of the cell. And then what you're seeing here is just different representations of ions that might move across this membrane to give us our change in electrical activity. And these little graphs that you see at the bottom represent the normal action potential in black, so that change in electrical activity. That action potential triggers a rise in intracellular calcium, which is this blue line here. And it's that calcium that's responsible for muscle contraction. So this dashed line here shows you how that contraction develops as that takes place. So from a physiology point of view, a heart muscle cell won't contract unless it's got an electrical impulse that causes it to do so. And when one of those impulses arises, it travels down this structure here called the T-tubule, which is basically just a pocket in the cell membrane. It means that that transmission goes deep into the cell very, very quickly. That electrical stimulus causes this channel here to open, and that channel there lets calcium through. So from the outside of the cell, through the calcium channel into the interior of the cell. Now, you'll see that there's a reason that this structure here is very closely linked to our calcium channel from a locational point of view. And that's important because that small amount of calcium that comes from outside the cell to in binds to this structure here, which is called the ryanodine receptor, which is another type of calcium channel basically, but it sits on that sarcoplasmic reticulum's membrane. And when calcium binds to that ryanodine receptor, that channel opens and that massive store of calcium that sat inside the sarcoplasmic reticulum can now exit and enter the cell. And that's what gives us our big rise in intracellular calcium. That binds to the filaments that cause contraction, and then basically everything gets reuptaken and pumped back out of the cell to restore low resting calcium concentrations. So this overall process is called excitation, contraction, coupling. And it's basically describing how that electrical stimulus is coupled to these processes that cause contraction. And that's happening on a cellular basis every single beat. Every time that cell contracts, all of those cells in the heart do at the same time, and that causes the heart to pump. So that's your crash course. That's several lectures in one, really, if I was to go into the full details, but hopefully that's conveyed enough that you need to know. Basically, an electrical stimulus gives us a small amount of calcium. That triggers a larger release from that internal store, and that rise in calcium causes contraction, and then that all gets reset on a beat-to-beat basis. So, our research question during my PhD was, here you can see some data where we're actually measuring the amount of calcium in the cell under control conditions, so normal conditions on the top, and then in heart failure underneath. And this region in the middle that you can see here is where basically we've stopped electrically stimulating the cell. So it shouldn't release any calcium. It shouldn't do anything from a contracting point of view. And what you can see is that in the control cells or in the normal cells, when we stop stimulating these cells, basically nothing happens. There's a little bit of noise. That's just a signal, background noise essentially, but nothing really happens. But in our heart failure cells, you can see that we get these quite regular, frequent, and also quite large spontaneous releases of calcium. So suddenly, for some reason, the cell is releasing a lot of calcium from its internal store. So we're wondering, well, what's the reason for that? And another thing that we saw at the same time was that when we looked at one of the ion channels expressed in these muscle cells, one called IK1, it basically just controls the movement of potassium. Control cells, basically that was normal. In heart failure cells, the amount of that IK1 being expressed was about half. So there was a massive reduction in the amount of that ion channel being present. So our first question was, why was there an increase in this frequency and amplitude of this spontaneous release? And was that reduction in IK1 the reason behind that? Third question was, could we explain any of that using a computer model that currently existed? And unfortunately, the answer to that was no, we can't, or we couldn't at the time. So existing models from a computational point of view weren't basically able to recreate this complex interplay of calcium. So they're what we call deterministic. If you think back to primary school, maybe, you might have used these number machines where you take an input, you put a number in, that machine has a fixed thing, which might be multiplied by 3, and you get an output from that. So if you put 2 into this equation, you will always end up with 6 coming out the other side. Deterministic basically means that you will always get the same answer from the original numbers that you put in. So these models basically couldn't handle that spontaneous nature that we're seeing up here. So then that led us to maybe we could be the ones that developed a model that could do some of this, which is what we did. Again, some complicated diagrams on screen here. This one on the left is basically the one that I talked you through earlier, showing you a cell and the different membrane currents and our sarcoplasmic reticulum friend here, etc. The one on the right is basically showing you a similar thing, but we're specifically modeling, or the schematic is showing you a much more complex way of recreating calcium movement through the cell. So basically, rather than having this one sort of like sarcoplasmic reticulum box here, we're replacing it with tens of thousands of these ones individually. So we've now incorporated a way that we can mimic and recreate some of that complexity. So basically, the bulk of my PhD was put these two models together and see if it works. Now, the specifics of all of that is quite boring. It's 4 years of my life that I won't get back, but it did actually lead us somewhere. And I'll talk about the end product rather than the kind of the boring specifics of all of that. So I did all that work. We merged those two models. Here are some graphs basically that show you that the model in solid lines closely reproduced experimental data. So that's in terms of the electrical activity, but also in terms of the calcium as well. So you can see how there's a close fit between the solid lines and the dashed lines basically. And we tested this across a range of frequencies, so basically different rates, just to make sure that we could capture a lot of the interesting dynamics. We then tested what we saw in those experiments. So we paced our simulated model, we paced our model for a period of time, and then let it rest and see what happens. And what you can see in the bottom here is the last of our normal electrical activity changes, the last of our action potentials. A little bit of time after that, you can see that nothing is happening, which is what we would hope, what we would expect, and that's what we saw in those control experiments. And then the video that I'm about to show you is a recreation of calcium concentration across those tens of thousands of individual units that I showed you in the previous diagram. What you will basically see is that there's one sudden bright flash and then it all disappears and nothing else happens. And that's basically showing you that calcium very, very quickly gets released and then gets reuptaken. And there's no sort of weird spontaneous activity happening. So here's our video. And that is the flash, and that is it. Nothing extra, nothing spontaneous happens. So I'll just play it again, just to make sure everyone saw it okay. So the model seemed to work, basically. It's a long story, short. But we wanted to explore that finding about that potassium channel a little bit more. So what happens if we simulate knocking that channel down like we saw in our heart failure cells? And basically what that did is it led to an increase in the overall amount of calcium in the cell, which is what you're seeing in these two right-hand graphs here, and a lengthening of the time that action potential takes. So if you look at this blue line here, it's longer than the purple one. And that's showing that the electrical activity has been affected by that knockdown as well. And when we put the model under what we call pro-loading conditions, so we mimicked adding a drug called isoprenaline, and that's the kind of thing that would mimic what happens when we exercise or sometimes in heart failure. What we saw was that the threshold for release of calcium spontaneously was exceeded. So basically, there is an upper limit to the amount of calcium that our sarcoplasmic reticulum can hold. And beyond that, it has to release it. It has to get rid of it because it can't hold it anymore. So that threshold was basically being exceeded in our knockdown of IK1 simulations. So then what we did is we basically ran hundreds and hundreds of different simulations in the same settings to see what happens on average every time that we do this. So what you can see here is basically the same as on the previous slide, that last action potential that we've stimulated and then a period of rest. And what you can see highlighted in blue is just, or the thick blue line is just one example of what that looked like in our heart failure simulations, and in purple, in our control simulations. So basically, it's still happening in control, but later on and not to the same extent. Now, this video that will come on next is the same as the one I showed you before, only what's happening in our heart failure condition. And what you will essentially see is that same instantaneous flash, but then we start to see some spontaneous events happening. And you'll see that actually they occur at two separate ends and meet in the middle. So this is a spontaneous release of calcium that's actually then propagating throughout the entirety of the cell. So there's our flash. Then you'll see some little pockets, and then you'll see from left to right and from right to left, we're seeing this spontaneous release of calcium that we're not triggering. It's happening because that threshold has been exceeded. And that's a bad thing, basically. Spontaneous release can mean things like extra heartbeats when there shouldn't be any. And that's one way that you might end up with an arrhythmia, basically. So our model gave us some new insight. We saw that these events were happening sooner and that they were bigger in magnitude. And we also saw that we did some extra cool stuff with this model, right? So something that we couldn't do experimentally, but we can do with the model is control the amount of calcium in the sarcoplasmic reticulum. So set it to a fixed value and force it to be released at the same time in both control and heart failure circumstances. And then see how that electrical disturbance, that change to that IK1 channel, is responsible for changes in spontaneous release. And basically what we saw was that meant that the spontaneous release led to what we call triggered action potentials. So full-blown changes in electrical activity across the cell, as you can see in blue here, happening despite us not applying any electrical stimulus to cause that to happen. And the reason for that is that calcium, as you can see, has a 2+ charge. So when it comes out of that intracellular store, it is raising the electrical profile of the cell. And in doing so, that makes the membrane more positive or makes the cell more positive, and that can potentially trigger these full-on extra action potentials that shouldn't be happening, essentially. And this was happening despite the cell membrane being slightly more negative than normal. You can't really see it very clearly, but the blue line is ever so slightly lower than the purple one here. And actually, interestingly enough, despite the amount of calcium coming out being less than in the heart failure condition, than in the control condition, sorry. So there was actually less calcium coming out when we did that, but it was having a bigger effect, is the moral of that story, basically. So, quick summary so far. I'm conscious of time, so I'll whiz through this. Basically, heart failure gave us a longer electrical action potential, and that led to spontaneous release. And that also destabilized our membrane and made it more sensitive. But this is all happening in a single cell. Some of you might be thinking, yeah, but the heart is an organ, so it's made up of all loads of different cells. So how does it actually translate to whole organ activity? And I'll come on to that. So another couple of videos here, another 3 videos, showing you in 2 dimensions now, so not just a single cell, a whole 2D patch of cells, what effect a spontaneous event can have on the electrical activity. So this video is going to show you the movement of an electrical wave from left to right, and then a spontaneous event is going to happen just behind it. And what you will see is that spontaneous event spreads its own wave of electrical activity uniformly outwards. So here we go. There it is. And you see that it spreads out nicely as a circle and then it stops. That's all that shows. Just go back to show you again. That's that. Now in the next video, I'm going to show you the exact same thing, only the time and the location of that spontaneous event originating has been changed ever so slightly, but you will see it has quite a drastic effect on how that electrical activity propagates. So rather than spreading out uniformly in one, it's kind of split off into these 2 waves that are spinning in opposite directions and constantly meeting in the middle, and they're continually going. They don't stop like the first one. They keep going. Otherwise, it's relatively uniform still. It's still giving us that kind of steady outward movement of electrical activity. And then the last one, again, same thing again, but slight change to the location and time of that extra impulse. And you will see we get a similar thing to the middle one, but then these individual waves start to break up and become chaotic, basically. Loads of individual ones start budding off, and we've got this really chaotic, random uh, electrical activity taking place. From an ECG point of view, this example on the left probably wouldn't really do anything too badly. We'd probably have normal sinus rhythm, so our normal regular heartbeat. This example in the middle, where it was kind of spinning but consistently, might give us something like ventricular tachycardia, which is just a very fast heart rate. Whereas this example on the right, the heart would lose its ability to pump properly. And we'd end up in a situation like ventricular fibrillation, which, if it's not treated, is potentially fatal, basically. And that's all from just ever so slightly different timings and locations of that spontaneous event taking place. And now if we build up the scale a little bit more, what you're seeing here is these two things that I've shown you before, those blue lines. So how a spontaneous calcium transient can trigger a spontaneous action potential, and how in the 3 dimensions that can potentially propagate across the whole heart. So here's, again, a couple of ventricles and that spread of electrical activity across them. Another series of videos down here just showing the same thing happening basically, but in the atria instead, so in the upper chambers of the heart, and you can see how these electrical wavefronts are spreading kind of chaotically and randomly, and how that's underlaid by changes to calcium. Now, the last couple of slides, all I want to do really is show you that it's not all about the computers. I may have made them sound like they're the fix to everything, but really, computer modeling and experiments go hand in hand. We wouldn't be able to do the modeling that we do without really good experimental data. So last few slides, I'll just show you how the two are linked together. Here is an example of an experiment I did during my PhD. So sorry for those of you that are easily grossed out, but this is a heart from a rat that's been attached to what we call a Langendorf apparatus. And that basically allows us to keep that heart alive, and it will still continue to beat if you feed it the right nutrients and stuff like that. But we can also flood that heart with fluorescent dyes that fluoresce at different levels when we shine particular light on them. And we can record the level of that fluorescence. And if those dyes are sensitive to electrical changes or to calcium, we can then actually record the electrical activity across the heart or the changes in calcium concentration across the heart. And these videos here show you some examples of what we would see. So this example on the left is a normal, regular pattern of electrical excitation. On the one on the right is an example of what tachycardia might look like, so an abnormally fast heart rate. So you can see how this wave in the left spreads out nice and uniformly and then disappears, whereas in the right, we've got this kind of spiraling pattern that seems to keep going around and around. So from those experiments, we can get data that look like this. This just shows us that spread of excitation across the heart and how it should be relatively uniform. And this diagram here just gives you an example of how we can use that spread to inform the models that we're developing. So that 2-dimensional spread of electrical activity informs how we might develop a model that mimics that exact same thing. So experimentally, that's called optical mapping. Basically, gives us that tissue level information on electrical stuff and on calcium handling. And then the last experimental thing I'll show you is a cool thing called diffusion tensor MRI. It's a measure of basically the fiber orientation. So muscle fibers orient themselves in a particular way. And diffusion tensor MRI basically measures the diffusion of water, which will change depending on the structures and the layout, basically, of a cell. So from this technique, we can get a measure of how the fibers, muscle fibers across the heart are actually orientated. And these are some really nice, kind of obviously exaggerated a little bit to make them look fancy, but examples of how the muscle fibers across the heart are actually arranged. And that gives us information from which we can infer how the structure of the heart influences the development of electrical activity. So again, a couple of examples under here where we're seeing spread of electrical waves across different regions of the heart. And these black lines that you can see in the middle here are basically just a mathematical what's the word? A mathematical way which we represent the center of this chaotic wave that you're seeing on the left. So where this actually originating from, and you can see how that is quite complex in nature, fundamentally. So the big deal is basically what we're interested in is how do these arrhythmias generate? How do they propagate? And how can we terminate them? So how do we go from the situation in the left here, where we have our nice uniform spread of electrical activity originating in our sinoatrial node, as it does in the atrium, to this chaotic electrical activity where that normal rhythm, that normal excitation has just been completely overridden. And you can see how in this example in atrial fibrillation, we've got this regular repeating sort of spiraling pattern that's sending off waves of electrical activity across the atrium. But it's all down to this lovely synergy that we have between experiments and models, fundamentally. So that is it for me talking. Thank you very much for your attention.